Welcome, everyone. Today, we are going to explore one of the most powerful and widely used tools in biostatistics and computational biology, the t-test. If you have ever wondered how scientists can confidently claim that a new drug works or that a specific gene causes a disease, you are in the right place. We will build this concept from the ground up. So let us dive right in. Now that we know our topic, let us look at a real-world scenario to understand why we need the t-test. Imagine you are a botanist testing a brand new fertilizer. You grow 10 plants with this new fertilizer and 10 with the standard one. After a few weeks, you measure them and find that the new group is, on average, 2 inches taller. That sounds great, right? But here is the critical question. Is that 2-inch difference a genuine, reliable effect of your new fertilizer? Or is it possible that you just happen to pick slightly taller seeds for that group by pure random chance? This uncertainty is exactly why the t-test was invented. Building on our botanist's dilemma, we need a way to separate truth from coincidence. The t-test acts essentially as a mathematical lie detector for our data. It is designed to tell us whether the difference we observe between two groups is a meaningful biological effect or just a random fluke. It does this by looking at two things simultaneously: the average heights of the plants and how much those heights naturally vary within each group. By weighing the observed difference against the natural background variation, the t-test allows us to make confident, scientifically sound decisions, rather than just guessing. Now that we understand the goal, let us formalize this in the language of biostatistics. The t-test is a specific type of inferential statistical hypothesis test used to compare the means of two independent groups. Whenever we conduct this test, we must always start with what is called the null hypothesis. You can think of the null hypothesis as the boring assumption. It is the baseline stance that there is absolutely no real difference between the two groups you are studying. In our plant example, the null hypothesis states that the new fertilizer does exactly nothing, and any difference we see is purely due to random chance. With our null hypothesis defined, how do we actually test it? The t-test calculates a specific number known as the t-statistic. This number measures how large the difference between the group means is compared to the natural variation, or noise, within those groups. If your t-statistic is very large, it means the difference is simply too big to be explained by random chance alone. We then calculate a p-value, which tells us the exact probability of seeing this data if the null hypothesis were actually true. If this probability is very low, typically less than 5%, we reject the null hypothesis and conclude the groups are genuinely different. Understanding these statistical concepts is not just an academic exercise. It is the foundation of modern biology. The t-test is arguably one of the most important tools in computational biology and medical research. Without it, scientists would constantly be fooled by random variations, leading to wasted resources and false conclusions. For example, when pharmaceutical companies test a new life-saving drug, they rely on the t-test to prove to regulatory agencies that their drug performs significantly better than a placebo. Similarly, in genomics, researchers use it to compare gene expression levels between healthy and cancerous cells, helping them pinpoint exactly which genes drive the disease. Now that we see how vital this tool is, let us look at the mathematics behind it. This is the formula for the independent two-sample t-test. Conceptually, it is just a ratio of the signal we care about divided by the background noise. Let us break down the symbols. The variables x bar 1 and x bar 2 represent the means, or averages, of our two groups. Their difference in the numerator is our signal. In the denominator, s1 squared and s2 squared represent the variance, or spread, of the data in each group. The variables n1 and n2 are the sample sizes. The entire square root term represents the standard error, or the noise. If the difference between the means increases, our signal grows and the t-value gets larger. Conversely, if the variance increases, the noise grows, shrinking the t-value and making it harder to prove a real difference exists. Before we move on, let us see this formula in action with a concrete example. Suppose we want to test if a new vitamin supplement increases the weight of laboratory mice. We are given two groups. Group 1 receives the vitamin and has 10 mice, an average weight of 25 grams and a variance of 4. Group 2 is on a normal diet, also with 10 mice, an average weight of 21 grams and a variance of 6. We want to find the t-statistic. First, we calculate our signal by subtracting the means. 25 minus 21 gives us a raw difference of 4 grams. Next, we calculate the noise by dividing each variance by its sample size, adding them, and taking the square root, which simplifies beautifully to 1.0. Finally, dividing our signal of 4 by our noise of 1 gives a t-statistic of 4.0. Because this value is quite large, we can confidently conclude the vitamin genuinely increases mouse weight. Now that we have successfully used the formula once, let us tackle a more complex example from genomics. A computational biologist is analyzing if the BRCA1 gene is expressed differently in healthy versus cancerous breast tissue. We are given 20 healthy samples with a mean expression of 120 units and a variance of 400. The cancerous group has 25 samples, a mean of 100, and a variance of 500. Notice our sample sizes are different this time. We calculate the signal by subtracting the means, giving us 20 units. For the noise, we divide each variance by its respective sample size, giving us 20 plus 20 under the square root. The square root of 40 is approximately 6.32. Dividing our signal of 20 by this noise gives a t-statistic of approximately 3.16. Since this is greater than the common threshold of 2.0, we conclude the gene is significantly downregulated in cancer cells. Having calculated the t-statistic mathematically, let us visualize exactly what those numbers represent. On screen, you are looking at two overlapping bell curves representing our two groups. The horizontal axis represents the value we are measuring, like mouse weight or gene expression. The peaks of these curves drop down to x bar 1 and x bar 2. The distance between these two peaks is our signal, the raw difference in averages. But notice the width of the curves. That width represents the variance, or the noise. If these curves were very wide, they would overlap heavily, making it hard to tell them apart, even if the peaks were separated. The t-test essentially measures the ratio of that horizontal distance between the peaks to the width of the bells. This diagram perfectly captures the physical interpretation of our formula. This visual understanding brings us to the ultimate purpose of the t-test. By comparing the difference between group averages to the natural spread of the data, the t-test provides a standardized metric. It is the critical bridge between raw, messy biological data and trustworthy scientific discoveries. Whether you are evaluating the efficacy of a new drug or identifying genetic markers for diseases, you cannot just rely on visual inspection or gut feeling. You need rigorous proof. The t-test allows researchers to make confident, mathematically backed decisions. Mastering this tool is an absolutely essential step in becoming a rigorous and effective computational biologist. We have covered a lot of ground today, so let us summarize our key takeaways. First, remember that the t-test is your primary tool for distinguishing real biological effects from random background noise. Second, it works by testing the null hypothesis, evaluating whether the difference between two independent group means is statistically significant. Third, the formula elegantly calculates the t-statistic as a simple ratio of your signal to your noise. Finally, a large t-statistic and a correspondingly low p-value give you the mathematical proof needed to confidently claim a genuine difference. You now have a solid understanding of the t-test, a cornerstone of biostatistics. Great job, and keep practicing these concepts.